Sir is currently serving as a research officer, scientist too, at the Drug and Standardization Department of Dr. Anjali Chatterjee Regional Research Institute for Homeopathy. This introduction holds a very special significance. I am Dr. Anjali Sharma and I feel truly privileged to welcome my mentor whose encouragement and guidance have helped me invaluably in my MD journey. With that, I warmly welcome you, sir, for the session today. Before that, may I now request the head of our institution, Dr. Shubhashish Ganguly, sir, to say a few words before we start our seminar with him.
Thank you, Dr. Anjali. Today, most welcome Dr. G.V. Narasimha Kumar, Research Officer, Pharmacology Scientist to DACRI, CCRH, Ministry of Ayush, Government of India. He is most welcome and we are very privileged that he has come to our institute for delivering and sharing his knowledge regarding the role of experimental pharmacology in homeopathic research, guidelines and recent advances. You know that 5 MD subjects are running in our institution, including homeopathic pharmacy, and experimental pharmacology is very important, particularly in relation with homeopathic pharmacy department, and various projects and research works are going on through our pharmacy department, and hope his scientific guidelines will enhance our department to progress the DND Homeopathic Medical College and Hospital in research and education. Most welcome Dr. G.V. Narasimha Kumar sir. Thank you very much.
Thank you, sir. With that, I hand over our stage to Dr. G.V. Narasimha Kumar, sir. Sir, please.
And mic.
Thank you, Principal Sir and staff for inviting me to this prestigious institute, and also Dr. Anjali. So whenever I visit a college, I recall my fond memories because I have started my career as an lecturer in a pharmacy college, teaching undergraduate, postgraduate students, and my interest towards, I was earlier doing my work mostly in experimental pharmacology, preclinical, but after 2 years of entering into my career, I was more into clinical sector. So I worked in Ruya Hospital, which is an government medical college and hospital in Tirupati, which is so much crowded, and I have worked in all the departments because even in our pharmacy we have a course called PharmD, where exclusively clinical pharmacology is being taught along with pharmacotherapeutics. So we also were going for rounds along with doctors and everyone. Then I got interested more into the clinical sector. Later I have moved into this government sector. So pharmacology, when I have come into homeopathy, I was astonished to know that pharmacology subject is not being taught in homeopathy, whereas in all the Ayush categories, Ayurveda, Draviguna, Siddha, Marutuvam, Unani, it has a name, Ilmul Udiva, Unani, pharmacy, nursing, MBBS, BDS, everywhere we have a pharmacology, because don't think pharmacology means it is about drug. It is how any medicine or any form of energy which modulates our physiological system. during normal physiology and also during pathophysiological conditions also. So how a medicine or any treatment works in our body by modulating. So we know that even homeopathy medicines work in a mild way, gently healing our physiological means restoring our physiological process. To my understanding, I may be wrong also. So these medicines also work one or another way. So how they are acting on, they will go on to the cell, because each cell is a structural and functional unit of our body. So these homeopathic medicines will also act on the cell. So with which part of the cell they are interacting? Cell surface, there are many receptors, or they enter into the cell, they act on ion channels, or enter into the cells, they act on our DNA. So which part they interact and bring about the changes in our body. So this is what mainly pharmacology we study about, or related to any medicine. Pharmacology in nutshell, we deal about two things. So when we take any medicine or any treatment, what our body does to that drug. Next, after that drug enters into our body, what that drug does to our body. So these two are the main areas which we will be discussing about. What body does to the drug is nothing but pharmacokinetics. Pharmacon means drug, kinetics means movement. So when we take the drug, it gets absorbed, it gets distributed in our body, then it undergoes metabolism, finally it undergoes excretion, ADME. This is called movement, pharmacokinetics. This is the first part of pharmacology. Next, dynamics, the name itself indicates, pharmacodynamics. The drug, by interacting with our body cells, organelles or particles, how it brings the changes within our body physiology system is nothing but pharmacodynamics. How drug acts on our body. This is called pharmacodynamics. These are the main two parts of the pharmacology, pharmacokinetics and pharmacodynamics. Experimental pharmacology is a branch of pharmacology. When we carry out an experiment and we study about pharmacokinetics and pharmacodynamics, this is nothing but, in simple language, experimental pharmacology. By performing an experimentation to study about how the drug acts on our body is nothing but, in simple words, experimental pharmacology. So coming on to experimental pharmacology, what does experimental pharmacology ask? So when we take a medicine, what is the most important thing? It's not the efficacy. The most important thing is safety. So from a drug development perspective, or a drug discovery or development perspective, only the drug which has been proven to be safe will proceed on to next stages of drug development. So in experimental pharmacology, first the researcher will ask the question whether the medicine is safe or not. So we carry out safety or toxicity studies in the first stage. If the medicine is found to be safe, then the next question comes, does it work? Does the medicine works on our body for the condition we are seeking to? So next step is, we will evaluate whether the drug is working or not. In simple terms, we do simple in vitro assays, in vivo experimental assays, model assays. For example, if a drug is taken for diabetes, anti-diabetic drug, we will take an animal model, we will induce diabetes in that animal, then we will give the drug and we will check whether the drug is acting or not, whether it is able to treat diabetes or not. This is the second step, does it work? Yes, it is working. We have seen that it is decreasing blood glucose levels or normalizing insulin levels. Then we come to the next part. How does it work? How does this drug work? This is nothing but mechanism. So whether it is acting on cellular level or by simple biochemical assays, or whether how it is affecting the DNA and altering the gene expression, or whether it is acting on hormones. So we delineate the mechanism. So how does it work? Next important thing is, what does it affect? The fourth question. This is a much more advanced next step, where we see what does it affecting. A receptor on the cell surface, an ion channel, or an hormone, or an DNA present within the cell, it is acting upon. So what does it affect? So what are the key pathways which is affecting biological pathways? So the omics part, entire thing. So what does it affect? So these four parts we discuss, we explore in experimental pharmacology. Is the first important thing is safety. When it is proven to be safe, then whether it works, how does it works, next what does it affect. These four topics we delve deep in experimental pharmacology. So in experimental research spectrum, so what are the different types of experimental models which we have? First, coming on to simple biochemical assays or simple chemical assays. So from that simple chemical assays, we will learn the physico-chemical properties of the drug. For example, you have gone through HPI. There will be presence of some test called when we take the drug, test for presence of flavonoids in a medicine, mother tincture, test for alkaloids, test for glycosides, what is its pH, what is its specific gravity, solid, total dissolved ratio, total ash, these things, physiochemical properties. These we learn by performing simple chemical assays. Next, coming on to in vitro, mainly cellular assays. We take a cell culture, we give the medicine, whether this medicine, how it is affecting the cells, those cells, cell viability we see, cell growth, cell division, cell proliferation, and we come to know how it is affecting the cells. Another type of model, whole, ex vivo. We isolate an organ from the animal or any isolated organ. We check how this medicine acts on isolated organs. Next thing, whole animal. We take a whole animal, give the medicine, and on a whole, in an integrated biological system, how the medicine is acting. This is another type of experimental model. Next, omics or system biology. We know we will be targeting by including computational systems, how this medicine might be acting, what are the key targets it is affecting, key biological pathways is affecting. It is more advanced part of pharmacology now. Next, in silico. It is a new trending one, in silico, where please keep in mind that all the in silico results which we are getting are simple predictions which needs to be validated in vivo. Whatever the in silico results, either toxicity or network or docking or molecular dynamic simulation, all these computational assays, they are predictive, not confirmatory. Those are they are simply predictive in nature. We need to validate those in the biological systems. So, in a nutshell, from molecular to cellular to organism to integrated biological response, next we get the translational. So, this is an approach now we incorporate in drug development, translational approach. And I would like to say that no single experimental approach will give us all the answers. There needs to be a mixture. One is dependent upon another. Using cell culture assays, we can high throughput. These are normally called high throughput screening assays. Means we have a like 100 medicines we need to test for a diabetes or hyperlipidemia. But we don't have that much time. If we test clinically, it will take years. But we need to generate some main medicines which will be having good efficacy. So by employing cell assays, cell culture assays, we can screen these medicines within a few days. Then probably 10, we can advance them into the animal models. From there, 1 or 2, we can advance into the clinical stages. So these, how we can employ this. In vitro pharmacology, as we discussed, mostly we do cell culture related studies. In vitro, we take a test preparations, take a specialized cell culture apt to the condition which we are trying to evaluate. Then give the medicine, measure their viability, their proliferation, their condition, and from there we will reduce the results. These, as we have discussed, these cell culture assays are controlled. What do you mean by the word controlled? Controlled means the temperature, humidity, the conditions of the experiment are controlled by us. In clinical, mostly we don't have a control over the patient. We have a limited control. But when we do preclinical experimentation, we have a control over the experiment. We set the what is the temperature in which the cells will grow, what is the humidity, what is how much CO2 will be given, for how much time the drug will be exposed to the cells, and after that we will evaluate. So we will give an controlled conditions and most importantly homogenized population. Clinically, patients are not homogenized. We will get a heterogeneous populations. Whereas in preclinical experiments, we get a homogeneous population. And most important thing, please note that clinically, we are treating a patient who has already disease which has been developed in him naturally. Whereas in cells or in animals, we induce the condition, we induce the disease. There is a major difference. So that's why there is a translational limitation also in this. That's why we are saying this is a control, but in a high throughput. Fastly, we can analyze the effect. Same in case of in vivo pharmacology. We take the medicine, and in animal models, especially the selection of animal is very, very important, because in animals also there are wide variety of animals. Mice, rat, rabbit, hamster, knockout models, nude models. The selection of the animal is an important one, because we are trying to, for example, screen a drug for asthma. The most suitable animal is guinea pig, because its bronchi are very, very sensitive. So we need to test those type of agents, mostly respiratory system drugs in guinea pig. Whereas we test if it in rat or mice, the results won't be appropriate or translatable. When we go for toxicity studies, we prefer rats, because rats are outbred strains, means they are a heterogeneous population. So based upon the condition, we will go for selection of the animal. And next, we can study, as the animal is a whole animal, we can study ADME followed by efficacy, pharmacodynamics and outcome. So it is a whole animal. So those parameters which cannot be captured. In case of a cell, we cannot capture ADME characteristics. So when we do cell culture experiments, we cannot capture ADME, absorption, distribution, metabolism, excretion. Whereas in whole animal, we can do those things. We can evaluate pharmacokinetics of a drug, even drug-drug interactions, which are very, very important. How one drug affects the action, T-half, pharmacokinetics of other drugs. It is very, very important. Based upon those things, and another separate part of pharmacology exists known as TDM, therapeutic drug monitoring, which we do mainly in ICUs, where there are drugs which have narrow therapeutic index, like digoxin, phenytoin, where a small change in dose will be lethal. So this TDM means the T-half or the action of the drug. If we are taking two drugs unintentionally, so what happens when we take two drugs, whether those actions are synergistic, antagonistic, those are very, very important, drug-drug interactions. These type of interactions we can study in whole animal studies, whereas in case of cells, we cannot study. That is the advantage of a whole animal system. So coming on to with this background, why do we need experimental pharmacology in homeopathy? So in homeopathy, we have a classical evidence, which is based on clinical provings. Whereas experimental pharmacology, we can do controlled biological experiments where the effect is quantifiable and evidence can be generated. So this becomes
and it is reproducible.
Reproducible,
That is also.
and it becomes a translational approach from bench clinic to bench means lab, which is a translational approach, and we can bridge these observations with an effect, quantifiable effect. So when coming on to homeopathy, the most important thing is we ask that whether homeopathy works. Everyone asks, but most important thing from a researcher, from a pharmacologist, we give specific importance to the test material. What we are testing, that is much more important, because if we are not sure of what we are testing, it is immaterial to ask whether it is working or not. We are giving an unauthenticated any drug and we are saying that it is not working. It is not fault of the system in which we are testing. The source itself is not authenticated. So the most important system we do in drug standardization in our council. So that is standardization of the material. So whenever we do any research in-house, we give importance to the source material, from whether we are taking and preparing a mother tincture, we will give keen importance to the plant, whether it is suitably identified, authenticated, then time of the collection of the drug, after that how it is prepared. For example, I can say you that last month we have procured Bellinis perennis plant for preparing a mother tincture from our institute in OT, and 50 kilometers from OT, Coimbatore, and also from Maharashtra, West Bengal. We have procured from 4 sources the same medicine, Bellinis perennis. So before proceeding on to preparation of mother tincture, we have sent it for testing the alpha toxin levels, pesticide content, and heavy metal. We found astonishing to know that out of 4 samples, 2 samples have high levels of exceeding aflatoxins. So if there is presence of aflatoxin, automatically the mother tincture prepared will be toxic. We cannot attribute the toxicity to the plant because inherently there is presence of alpha toxins. So while preparing the medicines, while we are before testing the medicine, it is very, very important to know whether that medicine is standardized, whether that is safe. Then only we can claim that yes, it is active. So please keep in mind that whenever you are going to do any research using any medicine, the source of the medicine, whether it has been standardized or not, batch to batch, whether you are using the same batch or there is a batch to batch difference, these small things we have to keep in note. So coming on to designing a rigorous preclinical experiments. So in preclinical, I will talk mostly related to animals which we are working on it. So we can correlate it to cell culture also. Okay. So mostly we discuss about animal experimentation. So for an preclinical experimental design, like clinical, it is somewhat correlating to clinical also. These are the most important things which we keep in mind. These 8 items are the one which we keep in mind before starting the experimental setting. First thing is research question. Whatever the research question I am posing, that should be doable. That is very, very important. And there should be a model, for example, animal model or cell culture model, able to do that. And the conditions within that setting should be feasible. For example, Dr. Anjali has come to our institute to do thesis work. While she has come, she had many ideas. But when we have sit on to the table, we have discussed about the feasibility. Because doing an experiment, not only technically, but also finance also matters. So there is a financial implications also. And the suitability of an animal model. So before doing an experiment, a research question is very, very important. We should pose to ourselves that whether whatever I wish to do, whether that can be done in that research setting, in that facility, that should be clearly distinguish before moving on to. Second thing is appropriate model. This is the most important thing which we look on to. Biological system which is relevant to our research question. For example, we need to do toxicity studies or safety studies. We need to take an animal model which is outbred in nature. Means animals, we do breeding. We have an animal house facility with in-house breeding. So animal are mainly breeding. When we breed the animals, it is of mainly two types: inbreeding, outbred. Inbred means we breed with the sister brother. So one mother has given the child, we next the siblings, we will mate between siblings. This is in order to induce homogeneity among the population. So for example, when we do an efficacy experiments to test the efficacy of any drug, for example, to test the efficacy of Syzygium jambolinum in diabetes, we should have a population, animal population with same characteristics to state that to know whether the results will be reproducible or not. For that, we need to take a homogeneous population. For that, we need to have a inbred. Whereas outbred, we mate between siblings of different parents. Means father, mother of one animal, female and male are different, which will produce a heterozygous population. In case of toxicity studies, we require a more heterozygous population. For that, we go for an outbred type of animal. So, selection of appropriate model. If we wish to do toxicity study and we take a homogeneous population, it will not, the results are not translatable. So, selecting a appropriate model system. For example, to screen any antihypertensive drug or anti-arrhythmic drug, dogs are very compatible and apt. Their cardiovascular system is 99% matched to that of humans. So we prefer beagle dogs for cardiovascular system agent screening. For respiratory system agents, we prefer guinea pigs. For pyrogen toxicity, we prefer rabbits. For normal toxicity test, we prefer rats. So selection of the appropriate animal model is very important. Then sample size. Sample size is also very important. In clinical studies, we have a large sample size, but it is not allowed in case of animals. But we also calculate sample size. Mostly the researchers, whenever they do, either they follow the previous studies or the right aptitude is calculating the sample size based upon the previous published articles, considering the effect size, significance p-value. We have number of softwares based upon which we can calculate. We normally use G-Power software to calculate the sample size also. Next, controls. Controls are very important. Controls, as we have studied, when we do any type of study, we take like in clinical study there are arms. In preclinical studies there are controls. So for example, we are going to screen an anti-tubercular agent for tuberculosis treatment. We keep a normal control where animals will be given only normal feed and water. They are normal animals. They will not be given treatment or they will not be induced with any disease. Next comes with disease control. In this disease control group, we induce only the disease. We induce tuberculosis. We will not give treatment. Next comes our treatment regimens, treatment groups or treatment controls, where we give the testing agents. Then comes positive control. Positive control, we need to compare our test medicine with an already established drug in the market. So in positive control, we take those type of drugs which are normally used currently conventionally. For example, for tuberculosis, we may go with rifampicin. Diabetes, we normally go with metformin. Antibiotic studies, we take paracetamol. And there are some studies where we do surgeries, experimental surgeries, also to induce disease. In those type of studies, we include sham control. Sham control means we perform the surgery and we will not give any treatment. For example, to induce peptic ulcer in an animal, we tie the pylorus portion. We cut out the stomach, means from externally. We tie the pylorus, then induce the peptic ulcer within them. So we do simple surgery. We will not give any treatment. Those are called sham control. So controls are also very important in case of an design of preclinical experiment. Next comes the dose or exposure. In case of homeopathic medicine, this is very important because in other systems of medicine, the drug mostly is in solid form, whereas in homeopathy, it is in liquid form. So how much dose, we will not say dose, we will say dose volume. How much volume of the medicine is being given. And most important thing is for how many days you will be giving this. Dosage regimen for a day, in a single day, how many times you will be giving. So how you will decide these things? These things will be decided based upon the disease or condition in which you are trying to evaluate this medicine. If you are trying to evaluate a medicine for pyrexia, fever, the treatment duration will be 3 or 4 days or 1 day. Inflammation 2 or 3 days. Diabetes is a chronic disease, so you will induce the disease and treat for a month. In case of cancer, it will take much more time. So dose and exposure is also should be decided prior itself. Next comes randomization.
So we have to standardize it.
Yes sir. We have to standardize, then we have to go into. Next comes randomization.
So how this standardization can be done?
Means duration or standardization of the model. For example, in case of experimental pharmacology, before any medicine is tested for any disease, for example, if we wish to screen a drug for diabetes, we have a standard model called streptozotocin inducer model.
That is a standard model.
Standard model. But before it becomes standard, the model should be developed and validated. So initially streptozotocin was taken. It was given from a 10 mg to 100 mg, 10 mg, 20 mg, 30, 40, 50, 60, 70, 80, 90, 100 for animals, for 1 dose, 2 doses, 3 doses. Then the scientists have seen, researchers, at what dose it is causing damage to pancreas without causing death.
No, madam is asking about the intervention. How
Means,
is
so how, like whatever you have told is with the modern medicine.
Yes, ma'am.
But here what we are doing is that we have a positive control of modern medicine. But how we select the dose for homeopathic medicine?
Dose, selection of the dose means
Dose.
dose volume.
Dose volume.
So selection of the
Even
dose.
the number of dose, the number of exposures. One is a volume, another is exposure. So how do we check that?
So in case of dose volume, in case of preclinical, we do safety studies initially before starting. As per OECD guidelines, we do acute toxicity study, subacute repeated dose toxicity studies. As per OECD guidelines, there are some fixed volumes. Like for example, 20 microliter per 100 gram, 400 microliter per 100 gram, 1000 microliter per 100 gram. We test in these predefined test volumes.
that is also a standardized.
Standardized, or in your clinical approach,
But
you
sir,
have
sir, that is in case of the drug substances which are toxic in nature.
No.
Nothing like that.
Toxicity studies means don't think that we are proving their toxicity. We are checking
sir,
for
means
safety.
I'm asking for those substances which we can take in the raw form. There are certain drugs also which are not toxic in their crude state. So then in that case, how do we select the dose of those substances?
That's what I am saying, madam. We will do a dose ranging study. This is also called dose ranging study. So for a certain period of days, for example, 14 days or 28 days, you will assume you will take 3 dose volumes, minimum, medium and high. You will be giving those to the animals for 14 days or 28 days. and you will see after the treatment period whether the at given dose are there any changes within the entire physiology of the animal. We will do like in humans, their hematological profile, biochemistry, their entire histopathology, and we will see whether they are safe or not.
একটা ডিজিজ মডেল স্ট্রেপ্টোজেরোসিস দিয়ে ডায়াবেটিস মডেল তৈরি হয় মানে তো সিজিজিএম মডেল 3-4 বলছে ওটা ওর তো ডোজ যেটা আছে 5 ড্রপস ভেহিকলস মে বি ডিফারেন্ট হোয়াট মেথডস ইজ তো ওটা দিতে থাকে ভলিউমটা দিতে থাকে কোন জায়গাটাতে গিয়ে ইন্টারভেনশনটা এফেক্ট আছে ওটাও মার্ক দ্যাট শুড বি স্ট্যান্ডার্ডাইজড
And I would like to highlight that 1 day of rat or mice is equivalent to 30 days of human. So based upon their normal lifespan is 1.5 to 2 years. Rat or mice, 1.5 to 2 years. So scientists, veterinary scientists, they have correlated. So based upon their lifespan and our lifespan, so how, because you will be saying, researchers will be asking, so we take anti-diabetic medicines for 2 years lifelong. Whereas you test for a diabetic medicine in rats for 1 month or 2 months. So how this data can be correlated? So this is how 1 day of the rat or mice is approximately equivalent to 30 days of human life, by which we will correlate the exposures also. Next, coming on to blinding. Like in clinical studies, blinding is also very important, because most of the researchers will be prejudiced to their medicines. Like initially when Anjali was also in our lab, she was saying, sir, curcuma longa is a good medicine, it should show effect. So I have told that we should not be prejudiced. We should give, we should know. It may come or it may not come, we should explore. So in order to be not to be, because in animal studies, all the mostly the parameters which we assess are subjective. It depends upon the researcher or the person who is monitoring. So they should be definitely blinded. And next comes predefined outcomes and statistics. Even the statistics, which is very important, like in clinicals, we need to earlier itself, what are the predefined measures or outcomes or parameters which we are monitoring, and what are the appropriate tests which we will be going to do. We need to be predefined. These are the 8 most important things in a nutshell, which is important for designing an rigorous preclinical study. And these 8 are being repeated in all the guidelines. Whatever we talk next in for few minutes, these 8 things will be repeated in those guidelines, because these are the 8 core pillars. Like we have discussed, controls. In a preclinical setting, controls are very important, because simple papers will be rejected, because we perform all the experiments, our manuscripts will be rejected at the stage due to not presence of an appropriate controls. In case of homeopathy, additionally we definitely require an vehicle control. Without vehicle control, we cannot justify our results, because that is the first question we get in case of our papers. So vehicle control is mandatory in case of our homeopathic medicines, because it is alcohol. And when we take mother tinctures, you should keep in mind, not only ethanol is a vehicle, the percentage of ethanol is also very important. When we take, for example, when we give mother tincture to animals, we should keep in mind what is the percentage of alcohol used during extraction. When we give potency, we should keep in mind what is the percentage of alcohol present there. But here there is a small glitch. In case of humans, normally we use the potencies as such in your clinical settings. But in case of cell cultures and animal studies, we have diluted those ethanol, because if you give 90% ethanol to any animal or cells, they will die instantly. In case of cells, even animals, you can find mortality. So dilution of those vehicle and also the medicine is also important. Next, reducing bias, like in clinical
So how
studies.
do we dilute?
Simple dilution, madam. We take the potency for example 30C, we dilute it up to 10% with distilled water.
So in both the arms.
Yes.
For the control also
Yes.
and in the
Yes. Because we have done the studies internally, because we are not sure at which percent of alcohol for giving repeatedly to animals that it is safe. So we have done a study giving from 10% to 90% alcohol. We know that 90, 80, 70 is damn sure it is not safe. But researchers they have a question: at what percentage of alcohol it is safe to be given to animals? Because basic research is being done on homeopathy like anything nowadays. But reporting, there is no standardization
yeah.
where how much percentage of alcohol is given. Someone is giving it at 1%, someone is giving at 20%, but we need to get to a point where the percentage of alcohol is safe without causing any harm to the animals. At which percentage we need to see the efficacy. In this point, there was a lot of debate within our council with experts also, like if you dilute the medicine, how can we say that
Yes.
it is?
Safe.
But we have asked them to think from our point of view, not from a homeopathic point of
So is
view.
there any guideline or is there any standard one?
We are devising that, because if we simply say that 5% ethanol is safe, up to 5% ethanol is safe, no one will agree. We need to show the data and everything, guideline. We are developing that. For that itself, we are doing experimentation on two sites to know whether the results are reproduced or not. So after that, we will publish it soon within 6 months, because it is much more important at what percentage of alcohol it is safe, and we can perform animal experimentation. Many researchers have asked us, if you dilute the medicine, it is not wholesome a homeopathic medicine. But there is no other way in case of giving to the animals. It is not like we are just exploring or investigating something, how it acts on animals. We are not giving a confirmatory result. Confirmatory result we will get from clinicals. But to know that we should have something in our hand, means we should be able to work with or tangible with. So we have defended like this, and they have agreed. So even in case of our preclinical experiments in homeopathy, reducing bias is very, very important. First thing is randomization. So for even while allocating animals to different groups, we follow randomization schedule, and the most important thing is we take the baseline characteristics of all animals. Means we need to say that all the animals, when they have been taken into the study, they are healthy, normal, both physiologically. Our veterinarian will be carrying out those physiological checkups, and not only that, we will be doing hematological and biochemical assays to say that yes, all the animals which have been included in the study, their baseline data is similar, plus or minus 10% is allowed. Next comes allocation, concealing. The medicines, they are being coded in our research studies also. It is because it is very easy to code in case of homeopathy medicines, because everything looks nearly similar except mother tincture, so it becomes easy for us. Next, blinding. So while we perform our studies, so our researchers are blinded until the data extraction. During the analysis of results itself, we are unblinding. Up to then, the medicine is coded. Next comes standardized procedures, which is very, very important. Standardized procedures for doing any experiment or any model, we should standardize all the procedures. What might be the standardized procedures? How we will give drug to the animal, at what time it will be given, how it will be given, how we will withdraw blood from the animal. We have a SOP. In what conditions the animal will be given, what you will do or we will do if there is any accidental death during blood withdrawal, how we will cope up with that. So these all things are standardized procedures.
These are in-house SOPs or there is a general SOP?
Madam, there are general SOPs, but we standardize it in-house. it differs from lab to lab because we cannot act exactly as such. We cannot adapt from another lab. It depends upon our conditions. And in case of animal experimentations, like a clinician, the
Sir,
hand
ट्रे
of,
पे रखो ना
चाय
ट्रे पे
लेंगे?
no, no, madam. Thank you.
बाद में होगा ना
ट्रे में रखो
Like a clinician handles the patient, the same way researchers handle the animals also. Their skill is very, very important. So when Dr. Anjali came to our institute for 2 months, she was just handling the animal. Because like a patient feels comfort in your presence, animal when it is taken into our hands, it should feel comfortable. Or else we cannot say that the effect was due to medicine itself. There may be the health, the physiological status of animals is also important. So the animal should feel safe and comfortable in our hand. So these all standardization procedures we follow before starting our study, and it is separate for each and every lab and animal house. They have to adapt and depend on their own. Next, what are the current guidelines or ethics which we follow? Before that, why do we need guidelines in preclinical research? Mostly in homeopathy research, there are mainly two things: to reduce inter-study variability. Second thing, to generate reproducibility. These two things are main. That's why we follow guidelines. I am doing a anti-diabetic study, and in your lab you are doing an anti-diabetic study. The results, we need to show that same medicine we are doing, and if we want to project that yes, in two labs it has achieved the same desired result, there should not be any inter-study variability, or to show that reproducibility, we should have a common guidelines. So we need to adapt some guidelines which is acceptable by the scientific community, not only in India, around the globe. So preclinical research has also few of these guidelines which needs to be followed while preparing the protocol, execution of the experiment, and also reporting of the study. So that's why we require even guidelines, and the guidelines are more stringent compared to clinical guidelines here. So these are the in nutshell recommended guidelines for legal and ethical requirements in India for running an animal house. We should have approval of CCSCA, Committee for Control on Scientific Experimentation of Animals. This is a statutory body under Ministry of Dairy Development and Fisheries. If we have CCSCA approval, then only we will be able to establish an animal house. And like IEC, we have a IEC here, Institutional Human Ethical Committee. Same way, Institutional Animal Ethical Committee will be there, and it will be constituted by CCSCA. And I would like to say that total 9 members will be present in IAEC, and 4 members will be directly nominated by CCSCA, and we don't have any control over them. We don't nominate anything, they will directly appoint. So they are very much stringent, and they simply look after the animal welfare. They don't have to do anything related to the scientific rigor or anything. They mostly look into when we are doing the experiment, animal welfare is being prioritized or not. That is the main priority of IAEC and CCSCA. Next, 3 R's: refinement, reduction, replacement. National Consortium for 3 R's in UK has developed these guidelines wherever applicable to reduce the usage of animals. Reduce, reuse, and refinement. Refinement is an important term. What do we mean by refinement? To provide the animals with necessary enrichments. For example, in this hall, we are listening, comfortably sitting in our seats. Animals we are keeping in the cages, which is not their natural habitat. They would like to run, they would like to play, they would like to interact. So enrichment is very, very important, and we take utmost care in that. We have enrichment items in our animal house, If you come, you can see we have a rat huts, mice huts, rat tunnels. We keep them in open cages, and we keep the animals in a group, minimum of 3, because they should interact among themselves.
So we have to provide entertainment for them.
Yes, sir. To maintain, to maintain their normal physiological status, and the food which we are giving should contain all the proteins, vitamins required for them. So this enrichment is very important. Even enrichment will be very, like, for example, I would say we have kept sir is present near the door. So 100 members are coming, opening the door and going. He will be feeling stressed compared to the person who is sitting backside. Same way, animals in a room, those cages which are present near to the room and far away from the room, they will be feeling stress. Those which are present near to the door, those animals will be feeling stress. Those which are far away, they will not be feeling that much stress. And next, the light intensity. So I am present beneath the light, I will have more stress. Whereas those far away, they will have less stress. So these are called confounding variables, which we also take care in our animal house. We change the cages daily, rotate the cages, so that the stress is also uniformly distributed among themselves. These minute things, we take care of them. Next, coming to planning animal experiments. Prepare guideline is available, where for planning animal experiments, protocols before planning experiments on animals. If we can go through this guideline, it provides a checklist which we have to follow. Next comes experimental design and reporting. Arrive is being followed in all the journals wherever you go. Conventional journals, they will ask us to stick to Arrive. Animal research, reporting of in vivo experiments. And toxicity studies. We follow OECD guidelines mostly. OECD, Organization for Economic Cooperation and Development. And bias and reproducibility. Like Cochrane risk of bias, we have this S-cycle risk of bias for preclinical experiments. And homeopathic preclinical methodology, we have 2 guidelines, REBAR and PREPOP. REBAR also reporting of homeopathic basic research experiments, which mostly combines with Arrive and Prepare. And PREPOP has been given specifically notion on the test article, homeopathic medicine, which needs to be standardized before carrying out basing experiments. So coming on to one by one, CCSA guidelines, as we have discussed, it is under this ministry, it is a statutory body. It plays a main role for, we require institutional animal ethical committee approval to start anything related to animal. We need to start an experiment, I should have IAC approval. We need to breed animals, I should have IAC approval. We are going to carry out a workshop in September to show the animals to the participants, to give them as demo. We should have a IAC approval, because those are not show pieces to show as such or give to any unexperienced researcher. For that also we need to take the approval from IAC. Animal procurement. To procure animal, we have an in-house breeding facility, we breed. But most of the institutions, they don't have the breeding facility. They will procure the animals for their experiments. For that, we need to have IAC approval, housing, husbandry, what is the temperature which needs to be set. If you come into our animal house, I would damn sure say that it would be clean and neat compared to our in-charge room. 24/7, 365 days. 21 to 23 degrees it will be set at any point of time. Humidity will be 50 to 70%. Light cycle will be followed, 12 hours light, 12 hours dark cycle. Clean, neat, odor-free chemicals will be used to clean the surroundings and tiles and everything. So these all precarious things will be taken into care as per the CCSA guidelines, because they will be having a surprise inspections. We have 2 inspections per year, which is not in our control. Nominee, CCSA nominee can come at any point of time and can inspect our facility. and they will give the inputs online. Most important thing, pain and distress. During giving sanction to any IAEC for any experiment, they will see how we are handling the animals, how competent we are enough to handle the animals, whether any procedure which we are doing causes pain or not. If it is causing pain, what remedies which we are applying, and next most important thing, whether the study has financial budget or not. In IAEC, we have Form B for application for experiments. We need to give the data related to the presence of financial allocation. Why? Why money is important? Because we start an experiment, we don't have the money, we didn't procure the feed. Animals are not being given feed. It should not be done. So they should have a continuous care. Veterinarian has resigned. So there is no veterinarian to take care of the animal. So our funding has not been there. Research personnel has resigned. So in order to take care of these things, financial budget approval is mandatory for giving an IAEC approval for any experiment. Human endpoints, they will see. Everything will be taken care by CCSA guidelines. To run a preclinical experiment, CCSA approval is mandate. Next, 3 R's. Replacement, reduction, refinement. Replacement. We will try to see whether the animal model is required, or we can do the same screening in a lower vertebrate like zebrafish, earthworm, nematodes, or even cell cultures also. If it can be done, we will go for replacement. If it is not feasible, the next important thing IAEC will do, reduction of animals. We have proposed 10 animals per group, total of 60 animals for my experiment. They will say statistically how many will be sufficient. 5 are sufficient. They will simply ask us to reduce it to 5.
But zebrafish also is good model for neurological disorders.
Yes sir, zebrafish is also, but those, for example, like carcinogenicity, like Parkinson's, in neuro also.
Right.
For screen these type of experiments, we will come to a whole animal. Next, refinement, which we have talked about, enrichment, which considers handling everything. It comes under 3 R's. 3 R's need to be taken care. These guideline needs to be taken care very diligently while performing any experiments.
Narasimha, I'd like to interrupt. What is your opinion about Innovo experiment, which is Innovo, which is very popularized by Dr. Satadal Das.
In vivo.
Innovo.
Oh,
The egg model.
Egg model.
Seeds.
so egg model. So egg model, which has a own limitations.
It is got some limitation, but
Limitations,
in case
but-
of angiogenesis and cancer, this model is
This
still,
model, we get preliminary results which needs to be definitely validated in an animal model.
so we have to validate it into a higher model.
Higher model, because
So,
by doing that in vivo model, we cannot exactly say that this medicine will be acting, because in vivo model we will not be able to induce whole disease, because it is also a single structural and fundamental unit, that is cell, and it is not interacting with other organs. So we need to go
it
after.
is very popular in virology department.
So popular, that depends upon their research question.
That's it.
Their research question might be suitable, so they have done. But anyway, after doing that in vivo, they are coming on to whole animal itself.
Because this is also in vivo experiment.
Yes.
And it is very cheaper and cumbersome.
Sir, when it is cheaper, it has its own limitations also. So, next, prepare guidelines. Prepare guidelines, planning research and experimentation procedures on animals, recommendations for excellence. You visit NORCOPA website, which has devised these prepare guidelines. You can find a checklist which you can download and go through all those things, which if you can be able to fill, you will develop a simple, good preclinical protocol. It is a ready-made one which we can download and look onto. What mainly prepare tells us? Prepare mainly like mainly focuses on interaction of the research personnel with animal house personnel, how we design the SOPs, and these all things it stresses on. Like P. P stands for research question and design, experimental design, and what is our research question. R denotes for responsible use of animals. We should not overuse the animals or underuse the animals, means sample size to speak effectively. Sample size should not be low, or else our money, time, everything will be wasted. Results will not be translatable, and the sample size should not be very high. Next, experimental procedures. Experimental procedures should be handled by a competent, skilled persons, and the pain and distress in animals should be kept in mind, because you are giving the medicine, but you are inducing pain to them. Then it will, even if you are giving medicine, it may not work, it may work or it may not work. It is very, very important in case of neurological disorder screening. Next comes project planning. We should prepare timeline. What timeline we should keep and milestones, and we should adhere to them. Next, animal welfare. Next, risk and resource considerations. Resources are very, very important and stressed by prepare guideline. Resources are nothing but not only funding, skilled research personnel also. If the research personnel who are dealing with the animal experimentations are not skilled, then the results which we are getting, we cannot say that they are translatable or validated. It requires minimum 2 to 3 months expertise to learn how to remove blood from an animal. When we go humans, we will easily remove through veins, etc. But in animals, we take the blood from retroorbital plexus, from the eye, in an anesthetized animals. That too quickly within 30 seconds. Because we give anesthetic agents which are randomly, which very fastly they will induce the anesthesia, and recovery is also very fast. Like inhalational anesthetic agents, isoflurane or sevoflurane. Induction will be very fast, within a minute. Recovery also will be within a minute.
So expertise is required.
Expertise.
So, one question.
Yes.
If we are giving drug, then if we are again anesthetizing those animals for the collection of the blood, so what about the interaction of that agent with the drug?
Yes, madam, I agree with you. So that's why CCSCA, which we have talked first, has given a list of agents which minimally act on the body. Like for example, this isoflurane, which are inhalational anesthetics, they act very quickly within that specific area. Their T-half and everything is very fast. They will not be present in the body for a long time. So that's why the
Same for our medicine, sir.
Yes.
Means in the ultra diluted medicine, their action, and that means, have you done any experiment to check this thing, that whether that has done any change with the animal without anesthesia? Because we cannot do. But the medicines, do they change the drug interaction, or do they react with the drug?
Madam, to carry out drug interaction studies in homeopathy or in Ayush system, it is bit difficult because we cannot tag the drug. So for drug interaction studies, we need to follow the drug. We need to take, for example, after giving the drug, we need to take the blood sample serially, time wise, and we need to check the amount or concentration of the drug in the serum or blood. So for that, we need to tag the drug with a fluorescent agent. Whereas in case of not only homeopathic medicine, Ayurvedic medicine, any medicine, there is not like a single compound for which we can tag, and we can attribute the entire action of the medicine to that single compound. That's why it is somewhat difficult in case of Ayush medicines or herbal derived. Whereas in case of allopathic medicines, paracetamol, I can tell you T-half and everything. For any phenatoin, those are compounds which we can tag. That is the main problem with that.
So plasma concentration and half-life is not available in our...
Yes, sir. T-half, ADME characteristics are not available. Next, most which is followed in all the journals wherever we go, they ask us to stick to ARIVE, Animal Research Reporting of In Vivo Experiments, devised by NC3Rs, If we go through this checklist, we can design and also execute and report the studies very diligently. The main Arrive has two set of lines. One is essential 10 and recommended set. These are the essential 10. One, study design. We need to present the study design. How many days, duration, dose, dosage volume, what are the groupings, everything. Sample size, we need to clearly present how we have calculated the sample size or how we have come to that sample size. Inclusion, exclusion criteria. It is very, very important. For example, we have induced diabetes. It is not sure that all the animals will get induced with diabetes. I have given streptozotocin for 60 animals, and it is not sure that every animal will get diabetes. So how you have included them? What is the relevant point where you have excluded the animals? This need to be given clearly. Randomization procedure, blinding, outcome measures have to be predefined. Statistical methods, experimental animals. We need to justify why these animals were taken. And next most important thing, sex of the animals. Because you might have read the papers, most of the papers they will blindly take the male animals. And they will say that we have excluded the female animals considering their menstrual cycle and hormonal irregularities. This is very, very common in case of basic research experiments. But NC3Rs and Arrive has said that wherever feasible, there should be inclusion of both sexes. Because whatever the medicine which we are taking, we are not giving exclusively only to males, except in case of some reproductive disorders. Those are being given to two sexes. But most of the researchers, to show reproducibility, they prefer for males. But this pattern is getting reduced because most of the journals, they are rejecting the papers which are discriminating this thing. They are asking us to include both the, and one more thing, why mostly males are preferred. When we breed the animals, here also there is a disparity. The number of pups given birth, male will be high compared to females. So for a breeder, when you approach a breeder for procuring animals, he says that I will be able to give male animals within 10 days. Female animals are very scarce. It will take 3 months. What researcher will think? I need to complete my experiment fast. So please give me males. Accordingly, he will put up in IAC. This is what practically being done. Experimental procedures. 3 R's should be kept in mind while we are doing the experimental procedures. And results. Next, this is the recommended set, which needs may be followed, but they are not strict on those. Abstract, we normally in IMRAD format, we give abstract, background, objectives, ethical statement, everything. Housing and husbandry. They are also asking precisely how you have housed the animals. Because for a cage with 210 square meter area, we can house maximum 3 to 5 animals. We can see in some papers they will write that we have housed 6 animals, but it is practically not feasible. And some persons will be housing single 1 animal or 2 animals per cage, and it is not also recommended. So housing, husbandry, everything, animal care and monitoring, veterinary care was given or not, how the animals were monitored, if unexpected death, that is attrition, has occurred during the study, how you have coped with that. These needs to be clearly presented in during reporting. Next, protocol registration. There are sites where protocols will be registered, but it is not recommended for a preclinical study because these are mostly exploratory studies. During the course of the study, we may add additional parameters also. So protocol registration, it is optional. Data access, we give entire data access when we publish our article for all the raw data and everything with supplementary information. Declaration of interest. So this Arrive guideline is very, very important because any journal, if we communicate for publishing and basic research experiment, they will ask us to fulfill the Arrive checklist. This is mandate. REBAR and PREPOP, they mostly being asked in Homeopathy journal and IJRH. Apart from that, no other conventional journals, they will ask for giving, adhering to ARIVE itself. OECD guidelines. These are mainly guidelines for testing of chemicals, not for any Ayush or homeopathy medicines. But their procedures, methodology is universally accepted. So we follow the procedures of OECD for carrying out the toxicity studies, where we adapted to them and we have modified the dose volumes accordingly. So we do mostly in our lab acute repeated dose toxicity studies. Apart from that, depending upon the medicine's indication and duration, we do genotoxicity, reproductive toxicity, developmental toxicity, carcinogenicity, skin, eye irritation, all types of toxicity studies. Next comes S-cycle related principle. Like Cochrane risk of bias tool, there is SYRCLE cycle, risk of bias tool for preclinical experiments also. These points also needs to be highlighted when we report an publication or a report in case of basic experiments. Like I discussed earlier, we need to address selection bias, we need to address performance bias, detection bias, attrition bias, and reporting. Normally in preclinical experiments, selective reporting we can find in most of the publications, where they will report only positive outcomes. They will hide or won't be reporting any negative outcomes. But we should report all the outcomes such that any other researcher.
Sir, in animal model, how the attrition rate can be there?
Madam, attrition rate, normally when we are planning the model, we will, based upon the model, we decide the attrition rate. For example, in case of like for carcinogenicity, anti-cancer drug screening, we can say that due to when developing cancer, there might be death of the animals. So plus 20% attrition rate we will calculate, and we will increase the sample size.
So that is related to the death.
Yes.
Okay. So means in patient we say that they drop out.
These
So
animals don't have the chance of
that
dropout.
is what I am telling. How the attrition rate can be there?
There is no chance of dropout. They should be included. But in case there is any accidental death, we keep. But that depends upon the animal model. There are some models we induce simple inflammation. There won't be any death. But in case of like we have induced epilepsy in animals, anti-epileptic drugs, we can see that we induce epilepsy by using a chemical called pentylene tetrazole. You give the pentylene tetrazole IP intraperitoneally, within 60 seconds you can observe petit mal, clonic, tonic convulsions, and within 100 seconds animal will die. Mandate. So for those type of models, we keep sample size high. And in those type of models, main outcome is percentage of protection from death. So this needs to be taken care. Next, coming on to REBAR and PREPA. REBAR, reporting experiments in homeopathic basic research. This is a 23-item reporting checklist which consists of mostly prepare and arrive checklist. Specifically, they are highlighting related to homeopathic drug. What potency, what dilution, how much volume. These, apart from that, remaining all the things coincide with arrive checklist. And pre-pop guidelines mainly related to potentized preparations manufactured according to current homeopathy pharmacopoeias. This guideline, this is not exactly a guideline, but a recommended principle has come in 2024, which mainly deals with standardization of the test reagent, that is homeopathic medicine. What is its source material, whether it is authenticated or not, how it was manufactured, what is its potency, dilution ratio, vehicle, batch, who is the manufacturer, everything. These pre-pop guidelines exclusively show the light or throw light on preparation, manufacturing of homeopathic medicines, which needs to be taken care before performing an basic experiments. So, in nutshell, what is the important distinction? So, each guideline answers a different question. So, when we take PREPA, when we look into pre-pop, it tells us how should research involving potentized preparations be scientifically designed and conducted. When it comes to REHBAR, it denotes how should the homeopathic, basic or preclinical experiments be reported. Arrive, it is in whole. How should the in vivo animal experimentation be reported? In case of CCSA guidelines, they are exclusively for animal house maintenance and care of animals. OECD, how should be the toxicity studies be carried out? So this is a clear distinction between guidelines. So when we are carrying out our homeopathic research, how to integrate these guidelines? So for preparing a scientific preclinical design, we should keep in mind both prepare and pre-pop guidelines. For carrying out safety studies, OECD guidelines, and for carrying out animal research, CCSA guidelines, IAEC, 3Rs and ARRIVE 2.0. For reporting and reproducibility, REBAR and ARRIVE and homeopathy test practical, pre-pop guideline is exclusively needs to be followed. So coming on to recent advances. So we know that from in classical pharmacology there were these assays, cell-based assays, animal-based assays, animal models, biochemical assays. But nowadays they are carrying out transcriptomics, metabolomics, proteomics, organoids, cell on chip, organ on chip models, in silico toxicity, computational models, hell of
So what is ex vivo?
ex
So
vivo.
means I am not aware of the thing,
Madam.
that's why I'm asking.
Ex vivo, we isolate an organ from an animal and we test the medicine on that isolated organ. For example,
Is it death or it is living? How can it be
we
isolated
will,
and be?
for example, we anesthetize animal or we euthanize and we isolate intestine. We keep it in chamber and we prepare all the nutrients, oxygen, everything to that
So that
tissue.
it can viable.
And we will give the
Nowadays,
medicine.
organelle is more popular.
We will give the medicine, we will check the intestinal motility, the contractions and muscle contractions. So these are ex vivo. Bronchi, we will isolate bronchi. We will keep the conditions as such which are present in vivo. We will give the medicine and we will say, we will record the
Who does
contraction.
this thing, sir?
We will. Pharmacological people, when we do our undergraduate, postgraduate studies, first we will learn most difficult studies are ex vivo,
That is
which
means I
takes
am,
from morning 9 o'clock to evening 5 o'clock. Tissue will be living, but to get results from that tissue, it is like a very tedious process because it is very delicate. We need to prepare all those things. It will be very, very simple procedure. Even
Similarly in organoid also.
yes sir,
So very
organoid.
precision of.
Very precision. Even we take, we go for an butcher shop, we bring sheep intestine and we perform experiments for our undergraduate students on that. We will not sacrifice the rat also. Sheep intestine, ex vivo studies. These all integrated, we get an integrated biological evidence. So nowadays trending one is network pharmacology, where for a medicine we get compound network target interaction, which are being published or shown in many and easy to do within a short period of time, requires only skilled manpower and computers, nothing more than that, and softwares. All those things can be done by using network pharmacology and systems biology. But it is a tedious process. At each step, we need to be very cautious, and after it requires an amalgamation of more than 20 to 25 softwares, and serially we need to go from one software to another software. So this is one image which we have done for Lycopodium clavatum medicine in the treatment of PCOD. So these are the green ones are the compounds and pink color hexagons are the pathways which these compounds are affecting, and these all 120 targets which these compounds present in the lycopodium is affecting the biological process.
Is it with the mothers or is it with the potency?
This one, madam, mother tincture.
Mother.
Because
What are the hub genes?
sir, hub genes like CYP19A1, INSR, PI3, AKT1, which are present in this like endocrine resistance pathway, pathways in cancer, apoptosis pathway. This is like a interlinked, and how these targets are linked to one another. These everything we get a concise report after we carry out network pharmacology. We will be able to tell our Lycopodium clavatum in the management of PCOS might be acting through this pathway and targeting or affecting or modulating these pathways. Once again, I am saying that these network pharmacology results are predictive, and this we need to. So before, for example, I have planned my PCOD experiment. I don't know what outcomes or what targets needs to be measured or screened in an animal model. There are many targets which we can target. So by performing a network pharmacology study, I will be able to deduce top 10 targets which the Lycopodium clavatum might be affecting in a PCOD patient. Then I will take those 10 targets and validate it in the animal model. It becomes an integrated approach, in silico guided evaluation of X medicine in the management of this disease. This is the approach nowadays we are following. Instead of blindly going on to all the pathways, we are narrowing our target selection with the help of network pharmacology, in silico guided pharmacology. So when we do network pharmacology, these main steps will be available. First, compound retrieval. For any plant or medicine, we need to retrieve the compound, prepare smiles, then we go for ADMET analysis. All these compounds which are ADMET, which are having good profile of ADMET will be selected. Next, they will go for next target prediction, then protein-protein interaction analysis, then network construction, followed by pathway analysis. Next, after that, if we wish, we can go for molecular docking, followed by dynamics and simulation. These all things we require more than 15 plus discrete manual steps, and it may take from days to weeks. So we were going through this, and for this we have developed in our lab this software which you can use for your research and everything, phytonet.ai. You can look onto it. So this will do all this work within few minutes. You can go into that and you can try that, phytonet.ai. It is an AI powered out of our own interest. So this network pharmacology is not being done only by homeopathy people. This is being followed everywhere. It is like a trending way. For example, when we publish a case, we not the thing. Doing the experiments with standardized procedures and which are reproducible by any another researcher in any another lab is important. For that, we require guidelines. We need to standardize our procedures and everything. So, this one we are doing an workshop. If anyone of you are interested, you can enroll for that, but similarly are limited. We have restricted for 15 itself because we don't want to take huge number, and because few people might be interested. We only want to cater whoever are interested, and no one should come forcefully. They should come out of their own interest and we want to invest our time also in those people whoever are interested. Thank you for your patience. Thank you for listening up to me. So if you have any doubts, you can contact me or you can ask me now if you have any doubts or queries, and you are free to visit our institute in my lab and animal house at any point of time with prior intimation. We will be able to provide you and showcase our facilities and everything, and we will also be able to help you in our research, and your inputs will also help in our research, because you are the person who are working on ground level with patients. So whatever we prepare the studies, mostly they are from the OPD. We take those conditions or problems from the OPD and we prepare those laboratory studies for which we need to do the work from the OPD. So thank you, and I
Sir,
will.
again
Madam.
I'm disturbing you,
Yes,
sir.
madam.
Sir, little bit about Phytonet AI. Little bit.
So this Phytonet AI, madam. So this is an AI based cloud agent. So you need to sign up. After signing up, you will get a free credits. For example, you need to give an simple uh, tag for that, simple prompt for that. For example, calendula officinalis in the management of wound healing. AI should have two main words, medicine and the condition. Please don't use symptoms, because in pharmacology it is being used in conventional pharmacology where as per ICD we need to give the disease. So Bryonia alba for Parkinson's disease. So this is the prompt. Then what this Phytonet AI will do means, it will search for the presence of biological active compounds of Bryonia alba through many sites, databases like Impact, Lotus, ChemBL. From there, it will extract all those active phytochemical compounds. And next step for this, all the agents, the machine will learn in smiles. For each compound, there is a skeleton which is prepared in presence of smiles. Smiles are nothing but, for example, alkaloid lycopodine is there. CC double bond CCCC carbon. It is their skeleton. That will be read by the Python language. That smiles will be retracted or extracted, smiles. After that, these compounds will go ADMET analysis. A compound will be favorable if it has favorable absorption, distribution, metabolism, excretion. From, for example, 100 compounds are present in Bryonia. Within that, only 90 have favorable ADMET. Those 90 will go to the next step. After that, target prediction. In our biology, in our humans, what are the targets in Parkinson's disease? Those targets will be retrieved from gene cards, drug bank. There are many sites. Background, it will. Then these targets, these compounds will, the next step, these compounds which were taken for Bryonia alba or any compound, they will be having targets. In the next step, targets of Bryonia alba and targets of Parkinson's disease will be interacted. Common targets will be taken out. These common targets then will be matched with the biological pathways in which they are present, and a protein-protein interaction image will be formed, PPI network. After that formation of PPI network, we will come to know, after PPI analysis, hub genes. What are the main hub genes which are being targeted in this disease? And what are the main top 10 compounds which are mostly affecting these top 10 genes? From there, then after that you can select. It is up to, after that you will get all the images like protein-protein interaction, the image which I have shown previously. After that you can stop, or you can select from the top 10 compounds, 2 or 3 or 4 compounds, and from the top 10 targets, whatever the target you wish to, and you can do docking. You can select and press the button for docking. It will do docking and will present the docking results. After that, you are not aware of the how to interpret this. At the last point, you will press the button, generate the report. It will generate the report in a publishable format,
Very nice.
which can be directly taken. So it's not like that we use it for.
So this is all related with the chemical constituents, not
Yes,
in the
exactly.
potencies.
No, because we can develop network pharmacology for any plant, only for plant medicines with presence of phytochemical constituents. It requires phytochemical constituents, or else we will not be able to do. Normally what we do, we can directly extract the chemicals from the databases, or we can do specific phytochemical profiling using LCMS-MS, which we have done in Anjali study. So ethanolic preparation like mother tincture, we will do specific LCMS based phytochemical profiling. We will export those compounds into this site. That has an option also. So it is two-way. Either you can extract the compounds from the database, or you can give the ones which you have found out after phytochemical profiling. This will generate the entire results in a publishable format, which can be easily understandable by any researcher, biological researcher.
So for my query, have you done anything with this lower potencies of medicines, or only with the mother tincture?
Madam, we have done with 6C also.
6C.
With, for example, in our PCOD studies for pulsatilla, lycopodium, we have
I
done
think Dr. Anjali's study you have reached up to 200, no?
200, sir. But for her, we have done mother tincture, because for doing LCMS also, it requires, we don't have the facility, and it requires finance implication.
That you require
Outsourcing, and after outsourcing also.
So, but this in say 6 or something, the chemical constituents in the mother tincture and the 6, I think it will be quite different.
Yes,
so
madam.
that is what I am asking.
Yes, that's what I meant to say. In the first step, compound extraction, either you can take the compounds from the databases which are already present, or you can do specifically your own study like phytochemical profiling by LCMS, where you will come to know these are specifically present.
Okay.
And you can export those constituents to this one, then it will take the constants from your exported data. Then it will perform, by which you can do for 6C also. For example, when we have done for pulsatilla, nigricans, mother tincture and 6C. In mother tincture, for especially ethanolic preparation, we have found out there were presence of 25 compounds. In 6C, we have found out that there are 20 compounds. And common there were 15 compounds, which have submitted to journal review. So we are getting up to 6C, whereas after 6C in 12C we were not able to find the presence of phytochemicals. Up to 6C
it crosses the
Avogadro limit. So up to 6C we can do the phytochemical profiling, and we can tangibly do with them.
Very good.
Thank you, sir. Thank you.
Thank you for your patience, whoever are there. So it requires a patience also to listen to anyone. My mentor used to say, a good speaker will be the one who keenly listens, because you no need to study, you just listen. Then you will be able to speak very knowledgefully. This is what, so that's why from our UG time, we were being taught that don't read books, just listen, go and play. You listen very keenly, then you can speak and you can put it on paper.
During Vedic period, Shrutis were there.
Yes.
Shruti and Smriti, both are very important.
So even if you don't read books, there is no problem. Practical, you listen to the one who speaks, the teacher. It will imprint the knowledge within your brain for a lifetime, and it can be delivered to anyone. The skill you can learn. Thank you.
Thank you, sir.
So at the outset, I would like to thank Principal Sir, Dr. Subramanian Sir, Madam, for inviting me here, and Dr. Anjali for facilitating my presence. Thank you. Thank you for.
Let's sit.
Thank you, sir. I'm sure your talk has given us a clearer perspective on importance of experimental pharmacology. And I'm sure each one in the house is enriched not only with just information, but also with a new outlook and renewed motivation to and perspective as well to pursue clinical research and preclinical research in homeopathy. So now I would like Dr. Shobhan Ghosh, sir, Professor HOD of Pathology and Microbiology, to kindly deliver the vote of thanks.
So on behalf of this institution, I'd like to convey thanks to Dr. Narasimha. Narasimha is a popular speaker and researcher, and he's a friend of mine for last 1 decade. This is very interesting discussion, today's discussion. पीछे बैठ के पहले बात होना था, लेकिन अभी चुप हो गया. That is very good. Interesting thing is we are discussing experimental pharmacology. It is really connect with the mechanistic of our medicine. It shows very interestingly, it is a precursor of clinical research. You know, we are very much popular with the clinical research in this institute, but preclinical research, which is a very fundamental one, so we have to well aware of the preclinical research also. So preclinical research or experimental biology or experimental pharmacology is talking about, which is the area where all the branches meets here. He's talking about the genomics, omics, that is the proteomics, genomics, epigenomics, and very interestingly, we have to learn one thing. That is a mother tincture. We are very interestingly, we can identify the phytochemicals. We can easily identify the characterization of the alkaloids or all the things. But when we are thinking about the higher potency, that is the potentization, succussion and the ultradilution, so we need more higher instrumentation like FTIR and Raman spectra. There are a lot of experimentation we need. And besides this, we have to prove 3 things. One is the biological activity, the biological signature in the animal. So that is very important. Sometimes science is asking from us, yes, we don't have any Avogadro's number in 1M, 10M, okay, fine. What is a biological activity in animal experiment? That is also very important. That gives you the answer from DACRI, that Dr. Narasimha is working for last 1 decade. Thank you very much. And his innovation is, I think, Phyto AI. You just, at least it is already hosted in the domain. You check it and sign it. And I think charges are very less. I think it's very less. In comparison to other sites, it's very less. Try it, and you are fortunate enough that you are already participating in workshop also. So that is a great experience, I think. So, last of all, on behalf of this institute, I again like to congratulate you, and really it's a very engaging session for all of us. Thank you very much.
Here I would like to say that this Phytonet AI, so we have a technical inputs. From my end, I have given technical inputs, but you will be astonished to note that the entire website has not been designed by any software person. It is done by my colleague, friend, Dr. Varaprasad, who is our research associate. He is a pure preclinical pharmacologist, but he has shown that if you have interest and zeal, you can do anything. By working on it for 2 months, he has developed, learned along with that within 2 months Now he is able to host this entire site. It's not an easy thing when you are a biology person and to design a website to put everything commercially and to hit host. It takes much amount of time because one step, it is a software. If you give different wrong programming, it may lead to. And I would like to say that this software has been validated by our colleagues, collaborators from Naipur and Zadavpur SRM. We have given them free module to validate, because whatever it is doing, it needs to be validated. We have internal validation in that. We have validated it tens of times, then we have released it, because the free hosting site, the question arises if the results are not appropriate or validated. So we have validated it and we have kept a nominal fees. This is not like for a business person. This for hosting rights we have kept the fee, and it is for whoever they are interested to know or to dwell into more into. Thank you.
Thank you, sir. And now, as we conclude our seminar for today, may I now request Dr. Shubhaman Ghosh, sir, Dr. Shubha Pramanik ma'am, Dr. Sangeeta Shah ma'am, Dr. Devinder Mondal sir, Dr. Shatyesh Naskar sir, Dr. Arpita Basu ma'am, Dr. Nasir Anand sir,